Next, I'm going to talk about high throughput sequencing or next generation sequencing. And there's a lot of different types of methods for this. I'm going to focus on the Illumina genome analyzer, so Illumina sequencing. And I'm doing this because that's the type of, that's what we have in our facility, but also it's like a pretty, highly used method. And it's pretty straightforward-ish and works really well. And so our facility has 3 different Illumina machines. We used to have an older version that we retired, but so I don't know, I'm not sure whether anybody particularly cares, but these are the guys we have. Okay, so the first thing that we need to do is fragment our DNA into manageable sizes. And so for high throughput sequencing, you're looking at sizes between like 100 and 150 nucleotides. And so we take our genome, we fragment it, now we have smaller pieces. We're going to ligate on some adapters, which are those specific DNA sequences. And so we'll put one type of adapter on one end and a different sequence adapter on the other end. And so now we have our genomic DNA, which is all fragmented, and each of the little pieces has these adapters on it. Then we're going to denature the DNA so it's single-stranded, and then we're going to attach it to a flow cell. And a flow cell is essentially a slide that we can attach the single-stranded DNA to. So that's what this guy is, and you can see it's single-stranded now. But it also has single-stranded copies of these little adapter sequences as well, already pre-attached on that flow cell. And so then what happens is once we attach our DNA, then the DNA can come over and base pair with the complementary adapter. And so you're going to form these little bridges. So and the next process we're going to use is actually called bridge amplification because of the way that this little guy looks when it comes over and base pairs with that adapter. And so now we're going to add in DNA polymerase, and the polymerase uses the adapter that was just by itself as a primer to amplify the complementary sequence to the original little piece of DNA that we stuck to the flow cell. And so after amplification, we've got this double-stranded piece of DNA. And now we can separate those 2 strands, so we could denature it. And now we're going to have 2 pieces of DNA that are complementary to each other. And now each of these are going to essentially repeat the process. So like this guy can come over here and form a bridge with that adapter. This one's going to come over here and form a bridge with this adapter. And we're going to just keep on doing this process. And so eventually what you end up getting are these little clusters, and each of the clusters represents the same sequence, right? So when we first put our DNA on this flow cell, we had, each of the little fragments of DNA could be a completely different sequence because we fragmented our genomic DNA in order to generate those small little pieces of DNA. And so, each of our guys originated as a unique sequence, but then after this sort of bridge amplification, we get clusters that represent an individual unique sequence where all of these are the same sequence. But there's kind of in 2 different orientations, right? So it's the exact same sequence, but some of them have the purple side down and some of them have the pink side down. And so there's a step in this process where you could remove all of the ones with the purple side down so that then, or pink side down, whatever, so that you just have the exact same sequence in the exact same orientation on your slide. And so then what happens? So then what we're going to do is the actual sequencing part. So up until now, we've just been making these larger clusters of our individual pieces of DNA. And that's really important for this next part, but we haven't actually sequenced anything yet. And so again, the sequencing relies on nucleotides that are fluorescently tagged. And so that sounds kind of familiar. In this case, the tag is actually removable. And not only that, but we're also using a nucleotide that has sort of a terminator, kind of like our dideoxy nucleotide that we talked about in the Sanger sequencing. But in this case, the terminator is reversible. And so we can use sort of a chemical process to make it so that this nucleotide can receive a new nucleotide. So you can't add another nucleotide to it until you want to, and then you use this chemical process, and then you could add the next nucleotide. Okay, so special nucleotides, they have a they're fluorescently tagged, and each of the nucleotides again has its own color. And then it's also, they have this reversible terminator so that you can't just keep adding, like let's say you have a sequence that's a run of like 5 T's or something like that. You're not going to just keep adding T's. You stop after every single nucleotide that's added until you allow it to go to the next one. Okay, and so then what we're going to do when we're ready to start actually figuring out what the sequence is we're going to add our polymerase, and we're going to add these fluorescently labeled nucleotides, and we're going to flow them over this flow cell, and then each of these sequences, the appropriate nucleotide is going to be incorporated, right? And only one nucleotide is incorporated, even if you have a sequence that's a repeat sequence of the same nucleotide, and that's because of that terminator that's on there. And so we do this, we flow this over there. We've got like each of these maybe has a different first nucleotide, and so we get a different color fluorescing from this. And we could take a snapshot picture of that. We remove all of this extra stuff, so this flows through, but then everything that's not incorporated is kind of washed off. We take a picture of whatever color is coming up for each of our little spots. Then we image that. And then we can do this little chemical method that's going to both remove the fluorescent tag, and it's going to remove that terminator, so now we can add on the next nucleotide. And so when we do that, then we could take another picture, and now that second picture corresponds to the next base. Then we remove, again, we do the chemical process to remove the fluorescent tag and allow for the addition of the next nucleotide, and we just keep doing that over and over and over again. Okay, so now we can line up all our images, and so let's say this was image 1, and then we had our second round, and we got image 2, then third round, fourth, and fifth. And then we could figure out what the color is at each spot on that flow cell, and use that to determine the sequence. So for example, in this location we have G, C, T, G, A, and then over here in this location we have C, T, T, A, G. And what's really nice, if we didn't have that reversible termination modification, then something like this guy, we wouldn't be able to tell whether there's 2 T's in a row or just 1 T or 5 T's, right? Because you just keep adding T's and it still fluoresce green. But that modification that allows us only to add one nucleotide at a time makes it so that you can tell, even when you have multiple nucleotides in a row, how many of those nucleotides are there. And so that's really useful. And so let me show you an image of one of these guys. This is a zoomed in image, and it's a surface image that's from one of these Selecta sequencing experiments. And each of the spots is a cluster of identical DNA fragments that was generated through that bridge PCR reaction that I was telling you about. And then the colors indicate which of the 4 bases were incorporated during that specific round of the cycle. The next step after you generate all the sequence data is actually to assemble the sequences. And I'm not going to talk about that in this video, but it does present a real challenge, particularly if you have these large genome sequences that have a lot of repeat DNA, not to mention that it's a huge computational load. And so that's something that is definitely an important topic, but we're not going to cover it. Okay, thank you for watching this video.